Hi, good afternoon, everyone. It's a pleasure to be here. Thank you very much. Okay, so today I'm going to switch gear to talk about a major Alzheimer's disease risk factor, APOE4. You have heard from Dr. Salko about Alzheimer's disease pathology, deposition of amyloid, tau tangles, and we know that the disease affects many different cells in the brain, not just neurons, but all the glial cells and the vasculature and so on. And obviously, the disease is multifactorial. There are many different risk factors. But I'm going to really talk about the genetics. So there has been, intense effort in genome-wide genetic analysis to look for genomic loci that can significantly change the risk of developing Alzheimer's. So today, there are, probably about, 100 at least different human loci that can show genome-wide significant influences in developing the risk. And on the left here, I'm showing you the Manhattan plot. And I want to point out that there are quite a few genes, or the genes that encode products that appear to play a role in regulating lipid metabolism. Okay. And as I said, APOE is the most important genetic risk factor for late onset Alzheimer's. On the right here, I want you to see I'm plotting the odds ratio versus the prevalence of each allele in the general population. And obviously, APOE, is the most significant here. You can see with one copy of the minor allele of APOE gene, you have more than threefold increased risk of Alzheimer's disease. And APOE minor allele is really also quite prevalent in the general population, anywhere between 20 to 25%, depending on the ethnicity. And then we have a whole bunch of loci that have very low odds ratio, barely above 1. So they can be more common, but they have very low kind of impact on increasing the risk. But then I want to also point to several genes here. They have very rare loss of function alleles, and they actually have quite a good odds ratio, about 2, so twofold increase in the risk. And if you look into the list of genes, turn out many of them, again, play a very important role in regulating lipid, particularly lipid transport, such as ABCA1, ABCA7, ATP, AB4. They all encode transporters that regulate phospholipid transport and cholesterol transport. So with that, with the genetics, I also want to remind you, in his original description of Alzheimer's disease pathology, Dr. Alzheimer, in addition to showing amyloid plaque deposition and tau tangle, he also noted very abundant lipid material accumulation in the brain. Okay, and then later on, I think now there are quite a lot of publications showing lipid accumulation in different mouse models of Alzheimer's disease. So the pathology plus the genetic factors prompted us to put forward the following hypothesis. So we think that the late onset Alzheimer's genetic risk genes such as APOE4, ABCA7, and so on, they causes lipid perturbations. And these lipid perturbations in different cell types lead to altered cellular states and function, and therefore lead to increased Alzheimer's disease risk and propensity for develop Alzheimer's-specific pathology. So now I'm going to focus more on the APOE gene. So I think many of you know that in humans, there are 3 major alleles of APOE genes known as APOE2, E3, and E4. And as I already alluded to earlier, that the APOE4 allele can increase the risk of developing Alzheimer's disease by at least threefold if one copy, and with two copies of APOE4, there are more than 10 to 15-fold increase in developing Alzheimer's. And then APOE2, on the other hand, is considered protective. So people carry APOE2 allele have reduced risk of developing Alzheimer's disease. And very interestingly, these different versions of APOE, they actually only differ from each other by one amino acid residues on either position 112 or 158. So it's the changes from cysteine to arginine. And this is considered to have a major impact on the folding of the APOE gene product. So what is the APOE gene product? It encodes the so-called apolipoprotein, and it is a secreted protein. Normally in the brain, APOE is the most highly expressed in astrocytes. And the function of APOE is thought to carry lipid and cholesterol from astrocytes to other cell types to support the metabolism, especially neurons. And on the other hand, APOE is also known to be the main component of the so-called lipoprotein particles, such as HDL. And such particles are known to be very important for lipid efflux. And APOE4 is considered to be defective in either binding to lipid or causing lipid efflux. So I'm gonna just show you some of the data generated over the years by my laboratory. So we became very curious to know how APOE4 impacts the different cell types in the brain. So back several years ago, we decided to do single-cell genomics in the postmortem human brain to specifically look at how APOE4 affects the different cell types. So we've been collaborating with David Bennett, who provides us with postmortem human brains from the RASMAP cohorts. And we also work closely with Manolis Kellis at the computer science at MIT to analyze the big data. So in this particular study, we curated a total of 32 subjects from the RASMAP cohorts. So we have 12 APOE33 subjects, 12 APOE34 subjects, and 8 APOE44 subjects. So we took their prefrontal cortical samples and conducted single nucleus RNA sequencing from these brain samples, and analyzed a total of about 160,000 single-cell transcriptomes. So here is a UMAP showing you all the major cell types that we recovered and analyzed. So I'm not going to show you all of the data. This is a lot of data to go through. But one point I want to make is that, so here on panel A, we look at differential gene expression between cells carrying APOE4 versus cells that don't carry APOE4. So then we organize the DAGs according to the biological processes or pathways. So I think immediately it became very clear. On the bottom here are the different cell types in the brain. You can see that APOE4 can influence different cell types differently. So the red represent increased gene expression, the blue represent decreased gene expression. So there are cell type specific increase and decreased gene expression caused by APOE4, okay? And of course, there are also genes that are globally influenced by the APOE4 allele. And I want to echo what Dennis just told you, APOE4 really play a very important role in amyloid formation and processing. And this function is conserved in many different cell types. But knowing that APOE is a gene regulating lipid transport and metabolism. We perform a second analysis focusing specifically on lipid and cholesterol metabolism pathways. And here, we also saw a very clear cell type specific impact of APOE4 in terms of regulating lipid metabolism. In particular, we became very interested in this profound upregulation of cholesterol biosynthesis genes in APOE4 oligodendrocytes. And on the right here, I just want to show you that the cholesterol gene activity is APOE4 dose dependent. So you see a rightward shift depending on how many copies of APOE4 these cells harbor. So another thing, just a point that, again, I want to drive home, which is APOE4 influence the same biological pathways differently in different cell types. So in astrocytes, what you see is actually a downregulation of cholesterol biosynthesis genes. Okay, so what's the significance of increased cholesterol biosynthesis? It turned out that there is an inverse relationship of increased cholesterol genes and downregulation of genes involving axonal myelination. So with that result, we immediately look to see, does that mean that APOE4 carriers are defective in myelination? And it turned out to be the case. So here we use several different kinds of microscopy to demonstrate this point. So, on the right here, you can see from transmission electron microscopy of the corpus callosum samples looking at non-APOE4 carrier versus APOE4 carrier. You can see the APOE4 carrier really show much thinner axon, myelinated axons. And on the bottom here, also just showing overall very disrupted axonal bundles in APOE4 carriers. So the question is whether this increased cholesterol activity really plays a causative role in defective myelination. So here we turn to iPS-derived oligodendrocytes. So my lab over the years generated isogenic APOE3 and APOE4 iPS cells, and then we can induce these cells into different CNS cell types. And here I'm showing you the comparison of APOE3 versus APOE4 oligodendrocytes and using Bodipi cholesterol to label the cells, and you can see immediately this APOE4 oligodendrocytes show very clear cholesterol accumulation. Okay, and we further perform lipidomic analysis and show that the species of cholesterol that accumulate in these oligodendrocytes are primarily cholesterol esters. So these are cholesterol attached to very long chain fatty acid, and they tend to be insoluble and tend to accumulate intracellularly. And we can show that this really is responsible for disruptive myelination, because we eventually were able to identify a small molecule. We actually screened for a number of small molecules, initially testing the various statin drugs, but the statin drug actually did not show very significant differences in cholesterol accumulation in APOE4 cells. So we eventually turned to a drug called beta-cyclodextrin. So cyclodextrin is known to solubilize cholesterol and promote cholesterol efflux. So we treated this oligodendrocytes, APOE4, with cyclodextrin, and we can show that this very profoundly reduced intracellular accumulation of cholesterol. And we also treated APOE4, humanized APOE4 knocking mice, and here looking at the corpus callosum axonal bundles, and we can see that this can restore myelination of these axons. So I think the series of experiments really demonstrated that there is a causal relationship of lipid dysregulation and disruptive cellular function. So based on that, a graduate student in the lab, later Kay, decided to further understand the regulation of lipid and cholesterol dysregulation in APOE4 oligodendrocytes. So she performed a genome-wide CRISPR screen looking for loss of function of genes that can influence lipid accumulation, either increase lipid accumulation or decrease lipid regulation, to understand the cellular pathways in APOE4 oligodendrocytes. And I just want to show you a couple of examples. They're pretty dramatic. So here, hit number 1, I'm showing you a loss of function of a gene that tremendously increase lipid accumulation in APOE4 oligodendrocytes. Okay, so you can see this huge increase of BODP accumulation. And on the right here, I'm showing you the opposite effects. Another hit, loss of function basically dramatically reduced lipid accumulation. So, so currently we are further working on these genes and working out their regulation, and we are also actually doing CRISPR screens in other glial cell types, including astrocytes and microglia. Okay, so so I have a couple of minutes left. I want to just give you another example of how we study APOE4 effects on microglia. So, so microglia are known to be the innate immune cells in the brain, but you know, I think there are emerging evidence also showing that microglia play an important role in supporting neuronal function. They basically can sense neuronal state and then respond in kind. So here, a former postdoctoral fellow, Matt Victor, established a calcium imaging assay to look at the activity of neurons and iPS-induced microglia in culture. So here you can see. So what he wanted to look at is how microglia respond to neuronal signals. So, so in this experiment, he exposed APOE3 or APOE4 iPS-derived microglia to neuron condition media, and then look at calcium dynamics. So on the top here, you can see APOE3 microglia responded very robustly, increasing calcium dynamics, but APOE4 microglia really failed to do that. They were not able to sense neuronal signals. So, so it turned out that this APOE4 microglia also are full of lipid. They show tremendous lipid burden. They upregulate a number of key lipogenesis genes, and then on the right-hand panel here, you can see this APOE4 microglia really show tremendous increase in lipid burden. And eventually, Matt was able to show that, again, if he is able to reduce the lipid burden in APOE4 microglia, then he can restore the calcium transient response of this APOE4 microglia to neuronal signals. So, so based on this result, the lab now has conducted a series of high-throughput small molecule screens in APOE4 iPS-derived microglia. We actually developed a genetically engineered assay that we can just directly look at lipid droplet accumulation. And again, we now have obtained a number of hits, and some of the hits you can see show dose-dependent decrease in lipid droplet accumulation in APOE4 microglia. So, so with that, I would just like to thank the people who have done the work in the lab, and I'm very happy to discuss our assays with you, and thank you for your attention.